Oh, I love your background.
Thank you. Yeah, if any postdocs are here, bargaining session, February 18th.
The name of it actually works. Yeah, that's downstairs. Yeah, but I don't work on biology. I actually work on it.
Okay, so it's just more, it's just math.
Statistics. Yeah, yeah. I mean, I really do like. Actually, no, that's all. I actually do like. Yeah, and I'm giving. Oh, hello. Yeah, I've seen your emails.
Yeah. A lot of people are like, oh my gosh, I know who you are. I was like, I don't know who you are.
Yeah, yeah. Yeah. You work at DSI.
For those of you watching there, there's pizza and there's also a side and sheet. So help yourselves. Yeah. So we just put the pizza on the bed and things.
Are there, is there going to be food?
There's a little pizza at everyone. Oh. Don't worry about it. And if there's more attendance, we'll be like, oh, you need to worry more stuff. Feel like what's the amount? Oh, so,
Yeah, because that's who I reached out to.
yeah, so so like I helped like make the material that you
Alright.
and I'm here to help people set up, or I'm here
For those of you who have your laptops here and would like to follow along with some code, I've set it up on GitHub, so there's an interactive link. So the URL here, a little tough to read, but my GitHub account is Diaz-Ale, Diazale. So I can, oh yeah, sure. So yeah, it's github.com/Diazale, and then my is it out?
Oh, they always are.
See, this is why we need chalkboards.
Not
Yeah.
I can't say that it is working.
Well, anyway, the rest of it is, you can see the URL, it's 2025 underscore DSCoV underscore workshop. Or you can just go to my GitHub, and if you navigate to my repos, I think it is the most recent one up there. So if you go to my GitHub, click repositories, there's this one right here. So if you go to that, at the bottom, there's a link here. It says launch binder. So you can clone this repo for yourself for your own research or any of your own purposes. It'll take a minute or two to load in the background. You may need to refresh it, but basically this will all run remotely. So you won't need to actually run anything on your computer. Your computer can't handle the data. So it should be okay. While that is running, I will actually be talking. So yeah, we can get into it here. So eventually it will load all of these notebooks and everything here, and you should be able to click into them and go and read through once they're set up. So yeah, it'll take I don't know, maybe like 5 minutes, but since I'll just be talking, you won't need to run any code or anything like that. So the first one we're going to start with is this one, Intro to Dimension Reduction. It's pretty much just text. And I originally did this for my PhD, so if there's a reference to McGill University workshops or something, that's what that is about. Also, just want to make sure, can people in the Zoom hear me? I don't know if...
Yes, we can hear you.
Okay, cool. Right, so first of all, everybody, welcome to this workshop. We're going to be talking about dimension reduction, also called dimensionality reduction, and its applications. And in particular, I'm going to be talking about visualizations. Why? Because it is really hard to perceive data in like 10,000 dimensions, but in 2 or 3 dimensions, it's a lot easier. So, what is dimension reduction in kind of the most obvious sense? You're taking high dimensional data and you're reducing it to low dimensions. So a really good example is something like a map. A map is a dimensionally reduced piece of information because you take a globe, which is in 3 dimensions, and you put it into 2 dimensions. So that's probably like the most canonical example. But if you also want to think about something that's a little more, I guess, theoretical, you can think about shadows. A shadow of a 3-dimensional object exists in 2 dimensions, and it tells us information about that object, but it's a transformation of it. It doesn't actually tell us everything. So on top of being able to get sort of a lower dimensional representation of the data, we need to understand that there's trade-offs. So with both of these examples, you can think of trade-offs immediately. So if you only look at the shadow of a chair, for example, depending on the angle of the light, you're going to see, say, the height of the chair really clearly, but maybe not its width or its depth or something like that. So that's an example of a transformation where you're going from 3D to 2D, and you're getting some information about, say, the geometry of the object, but you're actually losing stuff about the depth. With maps, we actually have a different sort of loss. See, we have a bit of a different trade-off because a globe is a non-Euclidean space. It's a sphere. And what happens if you peel a globe to create a 2-dimensional map, you're going from a non-Euclidean spherical coordinate system to the Euclidean plane, the 2-dimensional plane. So if you can imagine, you have a globe, you peel it off, immediately you've already lost information because suddenly, the oceans don't connect or something like that. On top of that, if you try to lay it out flat, you have to tear it because you're trying to make a rectangle or a square or something like that. You're trying to fit it onto a piece of paper. It's not going to work. So you have to make trade-offs. You have to make sacrifices. So you have to decide, how am I going to represent this high dimensional data in a low dimensional space? And if you're making a roadmap or something like that, you might want to preserve distances at a very small scale that reflect local distances. So that way, someone who's reading the map goes, okay, if I go 100 miles, I know I'm going to end up in the correct city. You don't really care about the distance from here to Calcutta, for example, because that's not super useful in a roadmap. If you're making a nautical map, you may be particularly concerned about preserving the oceans and making sure that you have the currents well represented. So there's different types of projections that are set up, and these are kind of different types of dimensionality reduction that you're familiar with, sort of intuitively through your real life. When it comes to a lot of scientific applications of dimension reduction, these are actually a little bit different. So if you've taken algebra classes or topology classes or things, or even statistics classes, you might have heard some terms like principal components analysis or PCA. You might have heard of things like neighborhood graphs, neighborhood graph embeddings, methods like UMAP, t-SNE, different neural networks, stuff like that. So these are all also used. So in this workshop, we're going to be talking primarily about three methods: PCA, principal components analysis, and my expertise is in UMAP, uniform manifold approximation and projection, which is a neighborhood graph method. And it's also related to another method called t-SNE, which stands for T-distributed stochastic neighbor embedding. These are all long technical terms. I have a lot of references in here if you're curious about them, but I'm going to talk about them more in terms of intuition and in terms of their strengths and their weaknesses, and also some applications. So out of curiosity, how many people here do single cell research or anything like that? So a few hands. Does anyone do genotype related research? So like population genetics, statistical genetics, biobanks, a little bit? Okay. And anyone like neuroscience or, okay, a couple neuro people. Anything else I may have missed? Okay, seemed like that was a pretty comprehensive set of options then. So why would you use dimensionality reduction in the first place? So I just want to make a note on terminology here. A lot of variables are referred to as features in the literature. So a feature could be something like the number of proteins, the number of gene expression counts, or it could also refer to the low dimensional variable itself that you've created. So that's just something to keep in the back of your mind. But why would you do dimensionality reduction? So first of all, reducing noise and pre-processing data is really important. If you have a computational pipeline and you have, say, a set of 20,000 genes that you're interested in, but you want to do some mathematical transformations on them, that's going to be a really big data set. If you're doing stuff like looking at genetic relatedness in a biobank, you're creating, I think, an n by n matrix. So, it's 500,000 by 500,000. You might want to reduce that through some manner. So reducing noise and pre-processing are probably some of the first applications we saw. In particular, PCA, principal components analysis, is really useful for this because for those not familiar with it, what you do with PCA is you take your data, which exists in your high dimensional space, and you transform it to a low dimensional space where the number of dimensions maximizes the amount of variance explained. So this is kind of a fancy way of saying that if you have, say, 1,000 dimensions in your original data, and you want to explain the most variance as possible in 5 dimensions, you would have to take the top 5 principal components. And that's just sort of a mathematical fact. You can do some linear algebra. Most linear algebra textbooks cover this at some point. But the thing is, in a lot of, say, machine learning pipelines, you might do principal components analysis first to reduce the number of variables from 100,000 to, say, 1,000 or 100. So that way you still get, I don't know, say 80% of the variation in your data, but you represented a much, much, much smaller data set. So that can speed up computation enormously, especially if you're mostly interested in kind of the roots of the signal of the data. So yeah, a really good pre-processing step. It can take your data down from like 1 million dimensions and bring it down to a few dozen and still capture a lot of the important signal that you're looking for. The other applications that I use it for primarily are visualization, exploratory data analysis, and clustering. So visualization is really, really useful. A lot of times, so my background's in statistics, and one of my favorite undergrad statistics professors, she always told me the first thing you should do as a statistician is look at your data. So we know a lot of ways to do this with like bar graphs, box plots, density plots, things like that. But with really high dimensional data, it can be really hard to choose what to look at. So if you have like 500 variables and they're all kind of represented by counts between 0 and 100, it's going to be really difficult to make 500 pairwise plots. You can use a method like PCA or a neighborhood embedding and visualize your data and then use that as a form of exploratory analysis. So this workshop is going to have some examples that look through that using genotype data and using single cell data. The other thing you can do is you can look for patterns. So clustering is a super, super common one. If you're looking at, say, healthy versus disease states of cells, you might have genetic data, something like gene expression counts, and you might run dimensionality reduction to see if there are clusters in your data. And you might see, for example, non-cancerous cells versus cancerous cells form different clusters. You could at that point say, okay, between these different clusters, what are some genes that come up more commonly? Or perhaps if you're working with human genetic data, what are some genetic variants that might appear in one cluster or another? So it's really useful. In terms of why would you do dimensionality reduction specifically in biology? So this kind of speaks to my background. A traditional statistical analysis starts with your hypothesis, and then you design an experiment, you collect data around it, and then you test that null hypothesis based off of the data that you've collected, and you come to your conclusion, which is really good if you have a hypothesis in advance, if that hypothesis is relatively simple, and also if you're the one collecting the data. That's becoming increasingly rare, especially with these huge biobanks or single cell data or neuro data. Often you go to an online resource like, I don't know, the Allen Brain Atlas, or you go to 10x Genomics or something like that. You'll download this huge data set. You don't know where to start. So, you've already kind of been thrown out of that original paradigm, which is referred to as Fisher's paradigm a lot of the time. Instead, we're often working within what is called Tukey's paradigm, which is named after John Tukey, a statistician who was really active in the 60s and the 70s. And the idea is that you start with the data and you make that the central point of your analysis. You visualize the data, and the idea is if you can visualize your data, you get a really, really good idea of what's going on in a way that statistical tests and tables might not. In biology in particular, this traditional Fisher's paradigm, you often assume that you had relatively few parameters and you had more samples than you had parameters, but still it was really small. It was pretty common to say I have 70 samples and I'm looking at 4 variables or something like that. These days, if you're working with the human genome, that's 3 billion base pairs. If you're working with a biobank, the UK Biobank alone has 500,000 individuals. So now your genetic data set is 500,000 by 3 billion. Like that's your original paradigm is completely out the window. You need something new. And the same thing goes with cells, where if you're doing single cell analysis, you're going to have, I don't know, 250,000 single cell samples, and you're going to have 20,000 gene expression variables that you're looking at. You can't really work in that original paradigm. So instead, what often happens is at the start of an analysis, you would do some dimensionality reduction, you would do some visualization, you'd look for patterns, you'd study, okay, is this pattern an artifact of my data? Is it something that I did to introduce a batch effect? Is it something really weird that's related to, say, like, oh, all the samples from one lab form a cluster, all the samples from another lab form a different cluster? You would start with this and, you kind of rule out all the technical aspects, batch effects, all these other things. And eventually you might say like, okay, I think there's actually signal in the data. Let's study it a little bit further. So there's tons and tons of examples in the literature of things you can do. So, you might find patterns while working with brain scans, looking at millions of voxels, and then you might try to correlate these voxels with gene expression. You might look at gene expression across embryonic development in C. elegans or Drosophila or something like that. I've seen some cool papers that do that. You might find subtypes of cancer given samples of cell surface proteins. So you might have a whole bunch of tumor samples, but the tumors might be heterogeneous even within an individual, within an organ. If you're working with a huge population data set and you're working with genotypes, you can find shared patterns of admixture and ancestry that can really kind of highlight like, okay, we see like there were migrations in the past that were kind of related to one another. Microbiome data, you can find all sorts of patterns, structure in microbiomes. So you could see whether there's natural selection going on in the human gut relative to the diet. You can do clustering in species to find subspecies. And also you can just reduce dimensionality for modeling and for computational efficiency. And I'm talking a lot about biology, but I've seen a lot of these methods applied in, say, digital humanities. People will take Wikipedia articles and they'll run dimensionality reduction, and then they'll find, different subsets and how they're connected to each other. So here this is how dimensionality reduction works. I've kind of already talked about the intuition about maps and chairs and things like that. But on the mathematical side, essentially what we're doing is we are just representing our data from a high dimensional space and a low dimensional space. And there's a couple different ways to do that. But generally what you're doing is you're minimizing some sort of loss function. So I don't have enough time today. This was originally a 4-hour workshop, so I'm not going to get into the details of the math. Don't worry, I've cut out some things. But the basic intuition is that, if you're looking at the two methods that I'm going to look at, or I suppose the two families of methods I'm going to look at are principal component analysis is the one that looks at global variation in the data. So you look at all of your data at once and you try to understand what is maximizing the variance in it. So was there a functional marker anywhere by any chance? Or is it... No? Okay, rats.
How about in front of your laptop? Like if you fold it.
Oh, this black one, there's another one.
I just want people on Zoom to be able to see it. An eraser. Okay. So I'll talk about it first. But basically the way PCA works, if you want to think about it a little bit more intuitively is if you imagine you had a cloud of points in space, what you're doing is you're drawing an ellipse around it. So in 2D, it's pretty easy to visualize. In 3D, it's also pretty easy to visualize. Think of like a balloon or something. In 4D or higher, just tell yourself 4 dimensions while you imagine in 3. Thank you. But the way it works is you draw an ellipse around the cloud of points, and then you draw the axes of the ellipse. And if you think about it, where the ellipse is longest, the longest axis, that's the biggest variation in your data. So that's your first principal component. The second one is the second principal component. The third one is the third principal component. So there's a lot of very technical descriptions that exist out there, but I think that one's the most intuitive, where basically what we do is we just draw a cloud around the points.
I got it.
Okay, thank you. And then we just try to use those axes. The other two super common methods these days are neighborhood embedding methods. So I'll just call them t-SNE and UMAP for short, so I don't have to keep repeating them. But the basic idea is rather than looking at the global variation in the data, where you look at everything at once and try to explain the variation, it's a neighborhood graph. So you take your data and you look for the nearest neighbors around every single point. Yes, it should be. Thank you.
Turn off your background.
Yeah, I forget how to do that, to be honest. I'm going to take a 30-second break here.
On the next to the video, the arrow.
It's the video, the arrow. None. There we go. And then, oh, right, because Apple has its own thing too. That one is going to be. All right. I'm just going to not use the marker for the moment then. But the basic idea is, let's say you have 10,000 cells in a data set or something like that. The way a neighborhood embedding method works is it uses topology. So you have these notions of nearness, of closeness, of similarity, of distance. And it provides a formal mathematical framework to measure that. So, if you think about distance, when we think about it, it's literally like, okay, that thing's like 5 meters away from me. This thing's like 10 miles away from me. And that distance, we kind of, grew up with it and makes a ton of sense. Another notion of distance is this idea of, okay, let's say you have 2 white blood cells that are different types of white blood cells and a red blood cell. Which one of those is closest to one another? So we have this intuitive idea of like, well, the 2 white blood cells are probably close to one another, like closer than the red blood cell, and they're definitely closer to each other than a skin cell or something like that. But how do we formalize that notion? Well, when you think about biologically, what defines a cell, I'm okay.
Oh, you're okay?
Yeah. When you think biologically, what defines a cell, it's something like the gene expression counts, the protein counts. So you might think, okay, so instead of talking about this sort of this like fuzzy hand-wavy notion of these cells are close to each other in some like subjective way, we find an actual mathematical measure. So for example, the white blood cells, they would have similar levels of protein counts. So we would say, oh, okay, so when these white blood cells are close to each other in the mathematical space, it's because the gene counts are closer. That being said, we have 20,000 genes that are being expressed, but they're expressed in different levels in different cells. So if in the high dimensional 20,000 dimensional space, we have similar gene counts, maybe we can find a way to say, okay, find my closest neighbors in that high dimensional space, the ones that just all have sort of similar gene counts. And then let's bring that down into 2 dimensions. So that way, when we look at a plot and we put those points on there, we can say, oh, okay, so all these points that are kind of near each other, they're close to each other according to the metric that we chose. And the metric in this case is something like gene counts. There's a bunch of other methods I won't get into, but CCA, canonical coordinate analysis, multidimensional scaling, diffusion maps, autoencoders, those are all there. They exist. Please look into them if you're interested. And finally, some cautionary notes. You should be careful when you interpret dimensionality results, dimensionality reduction results. The reason for this is because you are sacrificing information. You must sacrifice information. That's basically a given. You have to do it. The question is, what information do you sacrifice? And the different methods will have different strengths and different weaknesses. So PCA will show you the global data set. It'll maximize the amount of variance that you explain. So the top 20 principal components, they explain they are the lowest 20 dimensions that explain the most amount of variance. But it can be very, very sensitive to outliers. So if you imagine, I told you about the ellipse, the point cloud. Imagine you have a nice, like, tight football-shaped point cloud. and you draw an ellipse around it, it's going to look like a nice tight football. Now imagine you have 2 points that are way the heck over here. For people on the Zoom, I'm like way over here. Suddenly, that original tight ellipse is going to be really, really long, but just because there's a few outliers over here. So the variation in your data is not really representative of the data itself. It's because there's 2 outliers. So PCA is sensitive to outliers. That's one of the big drawbacks. These neighborhood graph embedding methods, so like UMAP and t-SNE, they're fantastically powerful for finding the local distances, for finding things that are super similar to each other. But you can't actually represent things that are similar to neighbors and different, you cannot represent everything that is similar to something and all the things that are different to it at the same time. And to put it a little more intuitively, if you think about it in terms of genetics, let's say you're talking about family members. So let's say you have grandma, grandpa, their daughter, and their grandkid, and the grandkid's dad who married in. So if you think about genetically, who are the people who are close in this family? So grandma, grandpa, daughter, and kid, all 4 of them are genetically related through the daughter. That, the husband or whoever who married in, he's only related to the kid. So that woman, she has neighbors, genetically speaking, with 3 people, whereas the man in the family, he only has the 1 genetic neighbor, and that's the child. So if you work in a bigger population data set, you may find that those individuals split up. And that's because, the man might have relatives in the data set that are not his own child. So you will be making sacrifices there. So if you've managed to, yes? Oh, does it only mirror tabs? Okay. Separate connection error. So for those of you who, binder not found. For those of you who clicked this binder, it should be loaded now. If it's giving you a server connection error, you can just reload it. And what we can do now is we can actually open up the toy examples notebook. So I'm going to run through this. Oh, yeah, it does Jupyter Lab now. But yeah, so we can run through some toy examples to give you a little more intuition, and then that'll take about 15 minutes. And once that's done, we can look at some real data. So I do have the sort of the mathematical explanation of PCA here. I'll skip it for the moment, but there's also a really good Stack Exchange thread that I archived like 10 times to make sure it's not lost to the seas of time. But it's got a really good discussion of what exactly principal components analysis does. But essentially what this notebook is going to explain, and you can either run it or I think the cell outputs are already generated, so you can just follow along. But what I'm doing here is I'm just generating random data, and it's randomly distributed in a way that it varies differently. So I took a 2-dimensional normal distribution. I think they both have similar means, but they have different standard deviations. So you're going to have it wider in one dimension versus another. So, this is what the data looks like here. Now, if you do PCA on it, basically what you're doing is you're drawing this ellipse around it. So this ellipse is kind of representing all of the data here, and you can see the axes. These represent the principal axes. So the longer one, that means the data tends to vary more along one axis, and it varies less on the other axis. So if you kind of generalize this in higher and higher dimensions, you can see how, okay, I've drawn this ellipse around my data, this ellipsoid in however many dimensions. These axes, they actually represent the most variance in the data. So the way PCA works is you just take your data and you transform it down to, say, the lowest 1 dimension or 2 dimensions or something like that. So essentially you generate your data, you transform it, you scale it. I guess the takeaway from this is when I was waving my hands, this is what I was trying to draw on the board. What we can actually do is we can work with some toy data. So this is real data from flower measurements from, God, probably like 100 years ago at this point, maybe 120. But the idea is we have, how many flowers? I think 150 flowers, and we've measured them across 4 dimensions. So we have the, is anyone here a botanist? Because I always forget what the sepal is. I think it's like a little stem that exists inside of the flower itself. But basically we have the length of that, we have the width of it, we have the petals length, and we have the petals width. So remember when I was talking about this intuition of like, okay, how are things similar to each other or different from each other? One of the ways we can look at flowers, for example, is, okay, they have similar petal sizes, they have similar seed sizes, things like that. So that's the high dimensional measurements. We bring them down to low dimensional spaces. So we have I believe, 3 species of flower in this data set. And what we can do is basically just use PCA to transform it. So this is the code here that will run principal components analysis for you. This uses sklearn, pandas. These are all really standard Python libraries. So if you ever want to run through it yourself, feel free to just use this. But I should mention one thing. The really, really nice thing about PCA is that you can figure out exactly how much of the variance is explained by each dimension of the principal components. So at the top of the workshop, I've got this equation here, but essentially, if you remember any eigenanalysis from linear algebra, the eigenvectors are associated with eigenvalues, and the eigenvalues in PCA, they represent the amount of variance explained. So what you can do is actually take the eigenvalues, sum them up, and then when you take the ratio of the, say, first eigenvalue to the sum of the 3, it tells you how much variance is explained by principal component 1. So in this case, if we take our 4-dimensional data set and we reduce it dimensionally using PCA, the first principal component alone now explains about 73% of the data. So that's a huge gain in efficiency. We went from 4 dimensions to just 1 dimension, and we managed to explain 73% of the data. The second dimension explains a further 23% of the variance. So that means with just the first 2 principal components, we have managed to transform our data. So it now explains like 96% of the data, 96% of the variance. So we went from 4 dimensions to 2, and we managed to preserve the maximum amount of variance in there. So a lot of times in a pre-processing pipeline, what people will do is, I think I might have a scree plot. No, maybe it's in the next one. Often what you'll do is you'll see a plot that, you just have the axes where you have the number of the eigenvalues, and then you have the variance explained, and it drops off. So typically you'll see the first like 10 principal components might explain like 90% of your data. And for a lot of people, they'll say, okay, well, for our pre-processing pipeline, we want just 90% of the data. And if it's 10 dimensions explain that versus 1,000, that's a huge computational efficiency gain. I'm going to talk more about visualization at this point, because that's actually what I enjoy doing the most. So the nice thing is, if you take your principal components, typically the top 2 are done, but any of the top ones, and you plot them just in a scatter plot of the coordinates 1 versus 2, it looks like this. And when you color it in, you actually see that clusters form. So in just 2 dimensions, you've managed to explain 95% of the data, and you see that these clusters are really, really clear. So we already know the labels because we have that in the data, but we can see that Setosa, for example, here, this forms a really clear, distinct cluster. These 2, they're kind of not as clear, but they still sort of separate side to side. So when I talk about cluster analysis, this is what I mean, where you look at your data, and when you visualize it, when you explore it, you go like, wow, okay, it's clear that there's multiple classes of data that I'm working with. This is not obvious looking at, this table of 150 values of 4 columns. So, you could still do it, but I think it would be a little bit more tedious. And, we are visual creatures, and barring any advances in like scratch and sniff technology, I don't think we're getting any better than being able to see those figures. So that's what PCA looks like using the iris data set. t-SNE and oh yes, sorry.
Is that the steeple length or things like that, or is that something else?
The components, they are their own variables. It's a pure transformation into the principal component space. So with PCA, you can actually do a back transformation, and you can figure out which variables are contributing the most to principal components. But just to be totally clear, the PCA values themselves, they represent a totally new space. It's a transformation of basis in linear algebra. So you're going from actual measures that you have into a space that explains the variation in the data. So there's some literature on like how to interpret principal components, and it is interpretable because it's a linear deterministic transformation. So you can just do the inverse transformations and figure it out. But like the actual components themselves don't have an immediate, they don't have a universal translation across data sets. And I'll show you in the next notebook that in population genetics, for example, there's a very cool sort of way that they represent data. And I think I might have had questions in the Zoom, but I'm not sure. Okay. So I don't know why my Zoom questions are not coming up. No, okay. All right. I saw notifications a while back.
Okay.
Yeah. Okay, thank you. Right, so this is one method of reducing the dimensionality of your data. So PCA, really cool, fantastic. But let's say you're less interested in the global variation in your data, but you're more interested in the local structure of your data. So rather than saying, okay, how does all of the data vary across everywhere? What if you're just interested in like, I want to figure out if there's any points that are kind of close to each other in a space. So we have these three flower types that we're looking at, these three flower species. Maybe there's a way to see three clusters more clearly, because if you think about this intuition of, okay, the different flower species, they're going to be similar to each other based on petal length or based on, I don't know, whatever other measures are in there. So maybe we can use that and try this approach of topological closeness. So in the high dimensional space, we're going to say, all right, with those four variables, we're going to look at, what are the 15 closest using a nearest neighbors algorithm. So k nearest neighbors is probably the most common one. There's various approximations you can use, but basically what you're doing is for every single flower in this data set, we're going to say find the 15 nearest points or the 20 nearest points or the 30 nearest points. You can optimize it however you want. And then we're going to do that for every single flower, and we're going to optimize some function that represents that four-dimensional space in 2 dimensions where we try to keep those neighborhoods together. So obviously, it's not going to work all the time because, if you have the 15 nearest neighbors, different points are going to have different 15 neighbors just because the way the data looks pretty much all the time. So you're going to make a sacrifice where perhaps the most distant points are not going to be as well represented, but the closest ones are better represented. So just as a brief note about t-SNE and UMAP and how they represent these distances, t-SNE, the reason it's got the T in its name is because it's for the student's T distribution. So the original algorithm was just called SNE, stochastic neighbor embedding, and it just approximated the distances in high and low dimensional space by the normal distribution. The sort of innovation in t-SNE was the low dimensional space is approximated by the T distribution. So that's not, there's a lot you can read on that, but it was basically just to make the visualizations easier. UMAP is sort of a like cousin of t-SNE. It takes a different approach. The paper is quite dense unless you happen to have a master's degree in category theory. So this link here is probably for the paper, so I wouldn't recommend reading it. Yeah, that's for the paper, but there's a how UMAP works explainer that's also written by the authors of the method. It's much clearer. But essentially the way it works is it just kind of does what I explained. It looks for the nearest neighbors in a high dimensional space, measures the distance, and then optimizes a function to bring it in the low dimensional space. This is it. So as an example of how these three methods differ, yes?
Could you maybe how what perplexity is in t-SNE and like how it affects the
Sure. So perplexity is right. Each of these methods have different parameters. So the one in t-SNE is perplexity, and perplexity is by default set to 30. It's kind of a weird parameter, but it is essentially kind of how small you want the neighborhoods to be. So the smaller perplexity is a lower neighborhood, and the larger perplexity is a larger neighborhood. So, when I said you want to approximate neighborhoods of your 15 nearest points or something like that, with UMAP, the parameter is just nearest neighbors. It's just n neighbors. So you can just say 15, and then it'll find the 15 nearest neighbors. Or you can set it to 30, it'll find the 30 nearest. For t-SNE, it's a little bit fuzzier, but it's kind of analogous. So like a lower perplexity means you kind of want the more local neighborhoods. That is a really good point to bring up because you may be interested in very, very fine scale structure in your data. So for example, if you're working with like single cell data, you might be interested in rare tumor cells early on in the life cycle of cancer or something like that. And there might only be 5 or 6 of them. So they will form a tight clump because their gene expression levels will be similar. Working with biobanks, if you look at the 25 nearest neighbors, that'll generally give you like overall genetic relatedness in the biobank pretty well. Whereas if you look at the 5 nearest neighbors or the low perplexity in t-SNE, you'll find like families get pulled out because they tend to be tightly related. So if you have a family that's mostly related to each other, and this is especially true in like some of the large multi-ethnic biobanks, you might have a population of like 30 people that came over together. in some wave of migration like 30 years ago, and they still don't have a lot of genetic relatives within, say, a country or something like that, but they're all relatively closely related. So if you set a higher perplexity or a highest neighborhood or nearest neighbors, you're going to end up sort of merging them with a larger data structure. But if you set it low, it's going to separate and form its own cluster. So in terms of how these three methods work, this is the Swiss roll data set. So it's just a data that's like, I don't know, like a fruit roll up, I guess. But basically it's just a three-dimensional coil plane, like a sheet of paper that's been rolled. The colors here are to represent its position in three-dimensional space. But if you run PCA, t-SNE, and UMAP on them, you'll see how they each represent data in two-dimensional space. So PCA, the colors here make it really clear what's going on. But if you imagine this didn't have any colors, it would not be clear at all. What the structure of this data is. PCA would just kind of say like, well, it's kind of a blob with a tail. t-SNE and UMAP, you can see that they still sort of unroll the Swiss roll and present it as this lengthy plane, which I think is more honest for the Swiss roll. And it's because it has a very non-linear structure. So PCA is a linear transformation. So these non-linear effects, they can really kind of catch it off guard. Now, the drawback, however, you see t-SNE and UMAP, they tear the data, because they're looking at local structure. So there's nothing special about this cluster here in t-SNE. There's nothing special about this cluster here in UMAP. However, the methods, because they only look at the local structure in the data, they will show you the most local structure. They might pull things apart because the points are like, there's probably like a floating point error or something here that moves a couple points a little bit closer. So it's just reflecting that artifact. So the takeaway here is that these points here, they are all close to each other in the high dimensional space. And it is represented as a plane. However, they're not separated. So the distance between these points here and these points here is not well represented. Whereas in PCA, it does represent the global variation of the data. So, you can see that these points here, they're this far apart in PCA, and they're basically this far apart in the original space as well. But you don't get the local structure of the data. Yes? Sorry, I'm hard of hearing. Could you speak up? Generally, you don't want to use dimensionality reduction for confirmatory analyses. Their biggest value, I find, is in exploratory analysis and also in diagnostics. So I found a lot of errors in data. I found batch effects. I found like significant regions of the genome that happen to really bias results. So things like that. So they're really good for the exploratory phase. For confirmatory, I wouldn't unless you really have a good sense of how your data works and also a really good sense of how the method works. So I would say this is a great place to start an analysis. Definitely don't stop with your visualization. Be like, okay, we did it. We found clusters. They're significant. So I have a little bit in the single cell notebook. But just to skip down a bit. So, looking at the iris data. So we have PCA on the left, t-SNE in the middle, UMAP on the right. And you can see the clustering structure is very distinct. And within the versicolor, I never read these out loud. I've used this data set for like 15 years. But there's versicolor and virginica. It looks like you can see the clusters do separate a little bit more. So in PCA, they're a little more stretched out, a little more intermingled, but they do get pulled apart a bit. So that's just to show how these work. So yeah, feel free to play around with those. You can generate your own data. You can run all of these cells in your spare time as well. I'm going to talk a little bit about genotype dimensionality reduction. So the human genotype so there's the genome, which is 3 billion base pairs. The genotype is a subset of that, and that's the one that tends to vary the most. So estimates vary, but let's say it's 1 million base pairs for this. We're going to work with real data. This is chromosome 22 of the 1,000 Genomes Project. So this is 3,450 individuals. We're taking their SNP data, the single nucleotide polymorphisms. And I believe the way the data is represented, here we go. So this is what the data typically looks like. So there's 4 values it can take on. -1 means it's missing. We don't have that. 0 it's when it's being compared to the human reference genome. So everybody, generally, you have 2 sets of chromosomes and then compare it to the reference genome. So you either have both chromosomes match the reference genome, or one of them is different, or both of them are different. So that's, there's other ways to represent it too. You can do 0, 1, 2, 3. So you could have like homozygous reference, heterozygous, non-reference, stuff like that. Or you can do just 0, 1, like same or not, but generally the results are going to be the same. So you can see here that even after working with just chromosome 22, just the genotype, we still have 11,468 base pairs that they're working with. This is really difficult to work with statistically. And one of the nice things that we have with the 1,000 genomes is we actually have the population labels. So it's 26 populations that were sampled from all over the world. I got to be honest, most of this code here is actually just setting up the visualization. So I'm not going to get too deep into it, but I do encourage you to look at this yourself because it'll give you an idea of how the visualizations are generated. And I've also got the code here to run PCA. So on my computer, it took about 0.7 seconds to run PCA and to generate the top 50 principal components. On this binder notebook, it might take like a minute just because it's doing it remotely. But you can see that with principal components analysis and the human genotype, so in this case, chromosome 22 for this data set, the first principal component, and this is the eigenvalue calculation I talked about before, the first principal component explains about 11% of the variation in this data. So we've taken our data from 11,000 dimensions down to 50, and that first dimension explains 11% of the variation in the human, in this chunk of the human genome. The second principal component is about 4%. The third one is, like 1.7, then 1.5. And after that, it really kind of plateaus at low values. So to give some interpretation to this, I'll come back to, actually, let me, hold on. Often what people will do in an analysis is this is called a scree plot, by the way, is they will create this plot and then they'll look for what they call the elbow, which is where you go from a lot of variance explained to relatively little variance explained. My PhD talked about how this is kind of useful, but also not necessarily great because you can have a lot of signal that's in, say, the top 30 principal components, but it's kind of arbitrary to just say like, okay, well, we're only going to take the top 5 because there's an elbow in the plot at that point. Which is just to say this is a cool way to look at how much of your variance is explained in the principal components, but you don't necessarily have to stop there. So a lot of journals and publications and manuals will say like, we chose this many principal components because we looked for the elbow in the scree plot, which is somewhat principled, but it's also somewhat arbitrary. So when you see that, don't take it as gospel. You can feel free to use more principal components or fewer. It's going to depend on your analysis. Right, so this code here is just running, this shows how to run t-SNE and UMAP. So all of this is preloaded. So I've generated it in case you wanted to, if you want to just load it, it'll be really quick. It'll take like a second. If you run it yourself, this is the code here, and it might take a couple minutes on the remote notebook. If you download it, if you clone it to your own laptop, it'll probably take on the order of a few seconds. But you can see to use t-SNE in Python, you specify the number of components. t-SNE can reduce data to 2 or 3 dimensions. I don't recommend using it in 3 dimensions. It's generally not very good, just because it tends to form like a big 3D ball that is not super explanatory. That may have possibly changed in recent implementations, but I haven't had much success with it. But the way that these functions work in Python is generally you specify the parameters that you're working with. So, t-SNE, I didn't specify the perplexity here. The default is 30. But you tell it how many dimensions you want to reduce it to, and then you run either the fit function or the fit transform function, where you take your data and it just does all of this for you. UMAP is really similar. It's got a few different parameters. So UMAP can reduce to an arbitrary number of dimensions. I find it's really good for 2 dimensions and for 3 dimensions to visualize an interactive space. You can also use higher dimensions for something like clustering or for pre-processing for machine learning. So when I cluster UMAP data, I often work in 5 dimensions or 10 dimensions. I find it's quite good. I have a whole paper on density clustering and dimensionality reduction if you're interested in it, and we can talk about that later. But basically, you see it's got the exact same syntax where you have, you tell it the number of components, and then you do this fit transform function. So in Python, pretty much all the dimensionality reduction methods are going to have this syntax. So skipping ahead a little bit, what does this data actually look like? So first we'll look at the PCA, and this is kind of a canonical result. And this is also, I should add, if you're excited about doing interactive plots, this code here will show you how to do it interactively. So it generates a little HTML file. So, you can click points on and off, you can zoom in. If this were a 3-hour workshop. We could go into a ton of detail on it, but instead I'm just giving you the code so you can do it yourself. But this is a canonical example of principal components analysis in genetic data. We get this triangle shape. So there's a few things I want to point out. You can see the colors here. They're kind of similar, and this is deliberate. The colors represent the, they're called the superpopulations in the 1,000 genomes, but there's basically 5. East Asia, South Asia, Africa, Europe, and the Americas. And those are just kind of like continental level populations. So the colors reflect that. And you can see that the colors kind of match here. So, you know, you have sort of African descended populations, European descended populations, East Asian, and then the green dots in the background are South Asian, and these pink dots, they represent the Americas. So when you think about, okay, what is the interpretation of principal components analysis in human genetics? What explains the most variation in the human genome? And the answer is basically geography, because the more distant you are from someone, generally the less likely you are to be related to them. And it's not for any super complicated reason. It's just that we tend to choose mates that are near us. That's just how it's happened for generations. It's been obviously a little different in the last few hundred years, but there is actually an interpretation of principal component space for human genetics. And it's called, there's a couple different measures, but one of them is isolation by distance. So if you actually, and there's a really cool paper by John Novembre, it's like the name November, but the French version. There's a really cool paper where you can actually take principal components analysis data and you can overlay it on top of a map of, say, Europe, and you'll see the points in PCA space from the top 2 PCs reflect the north, south, and east-west axis. So it's a very interesting result. And PCA is fantastically useful in population genetics, not just because of that, but because you know, you can see, oh, there's obviously structure in the data. So if you're running a genome-wide association study, you may want to use principal components as a variable in your linear model, because it would actually control for some of this population structure that you have. So this would get rid of things like there's this sort of deliberately obtuse analysis for the chopstick gene, where people did a what they did was they went base pair by base pair along the genome, and they found, oh, we found the genes that are causal for being able to use chopsticks, and they correlated exactly with having been born in East Asia. However, if you use a linear model that basically accounts for geography using genetic data, that correlation disappears because it's now part of the model. All that aside, there's a few things I want to point out here. First, you can see that these pink dots, they tend to appear in between sort of these terminal axes. And if you know a lot about the history of human migrations, it kind of makes sense, because if you think about Central America, South America, the colonial history, so obviously you had indigenous populations that lived there, then you had the transatlantic slave trade, you had European colonialization. So these individuals, they have recent ancestry from all the different continental populations, and that's just because of the waves of migration. So in principal component space, they tend to appear between clusters because they tend to be related to both of them. The other thing to note is these green dots. These are in the background. These are the South Asian individuals. And what happens is they actually fall along the 3rd principal component. So they're in the background. But in principal components analysis, we generally only look at 2 dimensions at a time. So if you plotted PC1 versus PC3, you'd see something similar, but the green cluster would form its own. But this also highlights one of the drawbacks of principal components analysis, in that you might have a lot of signal in PC3, 4, 5, 6, 7, 8, 9, 10, but it's hard to visualize because you would have to look at, say, 7 pairwise plots or 20 pairwise plots, and then you have to pull apart the signal. This is where you get the strength of methods like t-SNE and UMAP. So if you run t-SNE on the same data, you see that the continents form sort of these big fuzzy clusters. If you take the genotype data, you see that they actually form these continental clusters again. So now you have the green blob is no longer in the background, but it's its own cluster. However, the drawback, if you'll remember, is that you can't interpret the distances between the clusters. So the fact that this cluster is super far from this cluster, meaningless. That is not interpretable. But the fact that this forms a cluster is meaningful, because if you'll recall, it means we're looking at the local structure of the data. And this is done on the, I believe this is just the genotype data. You can also pre-process the data using principal components. I don't know if I've done that here. Yeah, this is the top. I don't remember how many of the top principal components it is, but I have a paper linked at the bottom that I wrote, and essentially using all the chromosomes, each of the populations actually forms its own cluster. And I have a whole discussion here on the interpretations that we have, and a couple other notes that you can look at in your spare time. But if you're interested in this kind of work with genetics, these 4 papers are a great place to start, especially the first 2, because I wrote them. So we have 10 minutes left. Time to learn about single cell processing. So there's a really common package called ScanPy. I don't actually use single cell research, or sorry, I don't do single cell research, but I read about it a lot because it's easy to steal methods from other fields and then pretend they're your own. So I'm going to talk a little bit about the differences between using a sort of pre-made package for dimensionality reduction versus doing it yourself. So up until now, we've just been doing it ourselves. We've been doing PCA using sklearn. And I think actually in the first notebook, we have PCA like straight from linear algebra principles. We've been using UMAP and t-SNE packages directly. But for something like single cell research, you might not have time to do all of that yourself. Like you have a lot of data you're working with. You have a lot of things you want to represent. There's like gene labels, cell labels. You might not like working with matplotlib. So there's a lot of packages out there that put everything together for you. So Python, one of the most common is called ScanPy or ScanP. I prefer ScanPy. In R, there's one called Seurat, which is very similar. So if anyone knows any art history, Seurat was, I believe, was famous for painting with dots. So I always found that was fun. And a lot of people seem to not know that. But yeah, in terms of, okay, should you use these libraries or should you not use these libraries? So the most important thing, which is why it's in bold, is always work to understand what your method is doing. Do not treat it as a black box. You should always know what it's doing. Like, these are just calculators. They make really cool figures, they do really cool things, but they're just calculators. For the same reason that you would never look at your calculator and just go like, well, I guess that's right. You should treat software the same way. You are ultimately the researcher, you're the scientist, you're the one doing the analysis, you're the one doing the processing, you're the one doing the interpretation, you're the one doing the writing. So you have to make sure that you know what you're doing. The same way you would not punch numbers randomly into a calculator and you would not use random functions, you should not just approach this as like, well, the computer told me this was right. But in terms of the advantages of these methods, prepared packages, they save you time. If you have a lot of experiments or huge pipelines or you're pressed for time because you don't want to learn like Plotly or something like that because it'll take 2 days, you can use it. If the packages are well maintained, you can avoid issues like dependency hell. So ScanPy is pretty well maintained. Otherwise, you might find like, okay, I'm in the wrong version of Python, but it's incompatible with dbScan, which is not compatible with UMAP, which is not compatible with matplotlib. Using the wrong version of Linux, my laptop's too old, my other laptop's too new. You can avoid that kind of stuff with a pre-made package because they tend to really be rigorous about that. Also, large libraries, they have active communities that deal with specific issues. So for ScanPy, you have a really big community specifically on single cell researching, which is really fantastic because I know Leland McInnes and Dimitri Kobach, they are developers respectively of UMAP and t-SNE, and I've seen their GitHub issues, and people are coming in with all sorts of like hyper-specific problems, and all they can say is like, well, I developed the method, maybe it's this, but like I honestly have nothing, I have no knowledge of this domain language. So the big advantage of a package like this is, the ScanPy community super active, the Seurat community super active. The disadvantage is people do treat them like black boxes. Don't do it. You know what your method is doing, and also take ownership over your work. If you do dimensionality reduction visualization directly, you control your parameters a lot more directly. Like you control the functions themselves. You also learn about the functions, and it can give you a lot of insight into how to do it. If I were to offer some advice at this point, I would recommend, especially with these notebooks, work with the methods directly so you understand what's going on. Once you think you have a good idea of how these different packages work, feel free to use all the prefab stuff because you don't know what you don't know, and it's not until you get into the guts of it that you can actually figure out like, oh my God, that's what this weird archaic looking parameter in ScanPy is doing. It's just changing the number of neighbors in UMAP or something. The last disadvantage is you can still end up in dependency hell, and it can be way more difficult to troubleshoot. So there can be a lot more things to address because you might have to say like, I'm using like these 17 libraries, and someone on GitHub is going to say like, oh, that sounds like a you problem. So yeah. Some other notes. This is a super active area of research. Like every 30 seconds, there's a new paper about like we've optimized local and global structure at the same time, which is a lie, but it's a fascinating one. There's a lot of debates on how to use dimensionality reduction or neighborhood embedding methods, whether you should use them at all. So I think the funniest series of titles is there was this one by Dimitri Kobach, The Art of Using t-SNE for Single Cell Transcriptomics, which was an explanation of like, here's how to use t-SNE to study single cell data, which was followed by Lior Paktor's sort of iconoclastic, The Specious Art of Single Cell Genomics, where he presented how it's like, oh, you can fit into an elephant. It's like, it's an okay paper. I think it's worth reading just to see some of the critiques. But it was then followed up by the specious art of seeing the elephant in the room. 2D embeddings of single cell data do make sense. I'm sure there's going to be another response in like 6 months. But you know, debate is good. A healthy debate is good and necessary for every field. If you do work with single cell data, you're going to be doing a lot of cluster analysis at some point. I would strongly recommend this paper, which is by, I guess, Anna Neufeld. I know the PI that was working on it, so Daniela Witten's lab. When you create clusters based off of genetic data, and then you analyze those clusters using the genetic data, you're doing statistically what's called double dipping. You're defining your populations by a variable, and then you're using the same variable for that analysis. Do be careful when you're doing that sort of stuff. So this paper goes into why you might not want to do that, but also presents some potential solutions. All right, in terms of actually doing this, so this is basically taken from the ScanPy tutorial, but it is, I just added a few extra details that I thought explained things a little bit more. But we're going to be working with PBMCs, which I always forget what it stands for. So it's some sort of blood cells, something mononuclear cells, peripheral blood mononuclear cells. I think that's what it stands for. But essentially the way it works is, you know, you basically import your data. So in this case, it's already pre-made for you as a tutorial piece. But the way that ScanPy works is it uses what's called an AND data object. So this object is a very, very simplified piece of code that contains everything you need. It's got the gene expression counts. It's got the labels of the cells. It's got you know, clusters that you have in there. It's got PCA coordinates, UMAP coordinates. It's all built into one object. So rather than having like 17 different things you're referencing, you'll just be working with this one object that contains all of your data. So if you want, you can take a look at this yourself on what's in there. I've given you the syntax for how to explore the package itself. But essentially what I want to get to is that, okay, you know, if you want to do UMAP just directly based off of your own understanding of how UMAP works, you can do that. So you create your UMAP coordinates and then you decide to plot it yourself. So in this case, I'm plotting, this was specifically for the gene counts for CD79A, which is a marker for B cell types. So you can see that if you plot the two-dimensional UMAP and then you color it in by the gene expression counts for this specific gene, this appears much darker, which means, okay, these cells, and they turn out to be B cells in the end, these cells that express the gene CD79A more, they are B cells. That's the type of blood cell, or that is the type of cell that they are. So if we go all the way back to the start where I talk about this intuition about nearness and similarity in high dimensional space, you have, in this case, I think 700 and something genes that you're looking at. And you have this intuitive idea of what does it mean that a cell type is similar to another cell type? Well, maybe we can take these 700 whatever genes, reduce them to 2 dimensions, plot them, and then we can color them in by all the genes and see kind of where they appear and get an intuition for which cells might be marker cells for a specific type. Traditionally, the way you would do this is you kind of have like candidate genes or candidate genetic variants in people, and then you would study them, you'd kind of go back and forth, and it's quite inefficient. This turns out to be a very efficient way to explore your data and to look at it. So if you wanted to just use your own plotting software, you would do it like this. However, it's already built into ScanPy, so you get the exact same result here, just using the plotting function, and you specify, I want to use UMAP, I want to use this data set, and I want to use this gene. So it's all built in for you, which is nice. And then in here, I've also got the principal components analysis. But basically, you can see, you know, the strengths and weaknesses of PCA and UMAP. So PCA here does create some separated clusters, but you can see the clusters are much tighter in UMAP. It's interesting because UMAP is not actually a clustering method, but when I spoke to the developer, he said his motivation was actually to find clusters in data. He worked specifically in, he works for CSEC, which is sort of the Canadian equivalent of the NSA, but he was looking at like clustering emails to find, you know, phishing emails or something like that. So it was really interesting to hear that application, but how it ended up being very applicable. You know, the advantages of ScanPy is like, okay, let's say you want to do like 10 variables at once. You just use this one function, it generates it for you. So here you have CD79A, MS4A1, IGJ. I'm going to continue reciting these as if I know what they mean. But then you have like counts, and then you have the bulk label. So what you can do is you can plot all of these 2-dimensional representation of cells at the same time. You can color them in by the gene expression values that they have. You can take a look at the labels that you know, and then you can just kind of see visually like, oh, I think this is worth exploring. Perhaps CD3 is super important in this type of cell over here. I guess that's not dendritic. I can't, oh, the CD4. Yeah. And then there's a couple other things. I haven't talked about clustering because that's a whole different issue. This is lead-in clustering, which is one method of clustering. Other methods include k-means, density clustering, which I'm happy to talk about, and I've written a few papers on that. But yeah, you know, it's all built in there. So that's like a super crash course on dimensionality reduction. Feel free to use these notebooks. Feel free to contact me afterwards or get in touch. Really happy to help with this kind of stuff. It's always deeply fascinating. I'm always interested in learning what people are using these methods for as well. So yeah, with that, happy to help out and take any questions. And also, if it was like super, I know there was a lot of material, but you know, if you have any questions on like even very basic things, happy to answer. Yep.
My understanding is like for PCA, you'll have like going back to like the plant where it's like you're comparing like petal length and width,
Mhm.
and then you're kind of seeing like what's the variation. And I guess my question is, you'll have like multiple of these comparisons, so then like plant width, petal width, and stuff like that. I guess my question is, how do you know for the different components? What if it keeps on being one factor in each when you do those, because each component builds up a comparison of two of the
Yeah.
factors. What if there's like one factor that is always part of like the components that are most explanatory? Like,
Yeah, so that does happen. The question is, I guess if I'm understanding correctly, the question is like, what if one variable just dominates the variation in your data? So
can you see that?
you would see that. So that's why it's, and I guess I skipped over this, but you really want to scale your data before you do it. So if you think about, you know, let's say, for example, you were looking at cars, because there's a cars data set in R that's really common. But, you know, what are some of the measures of that you could have for a car? You have its weight, you have its horsepower, you have its, I don't know, like max speed, fuel efficiency, things like that. You know, if you put its weight in grams, that's going to be a huge number, as opposed to like number of cylinders, 4 or something like that. So the idea is you want to scale and normalize your data, and then you would do PCA on it.
I'm relatively new to like I'm right now learning like the spatial transcriptomics.
Yeah.
And I guess one of the things I was wondering is, so oftentimes they use, I guess I'm kind of confused why clustering. I know you talked about the egg clusters around neighbors.
Yeah.
I guess why, like, especially like in this example with like the Swiss you showed.
Oh yeah.
Like, I guess I'm kind of confused what's happening to those, like when like the PCA versus UMAP wise, what's happening to the things, or is it just points or? Yeah, I guess I'm kind of confused what happens to everything. What, yeah.
So if you want to think about it this way, the way UMAP and t-SNE work is they find the neighbors. So let's say you're looking at this red point here. It's finding the nearest, say, 15 neighbors, and it's building a neighborhood around it. And then it's doing that for the second point, and it's building a neighborhood around it. And it's doing that for all of these points. So the way UMAP and t-SNE represent data is, okay, we have this red point, we're going to rebuild the neighborhood in the low dimensional space. We have this red point, we're going to rebuild the neighborhood in the 2-dimensional space. Same with UMAP. Now, because these neighbors are all really close to each other, and they're closer than these like yellow-green points over here, these yellow-green points are just going to be lost. They're not going to be included in UMAP or t-SNE, which is why you don't see it pulling towards this part here. PCA, on the other hand, is just explaining where the data tends to vary the most. So, you know, the axes run between basically -10 and +10. And it's not super clear which axis, like this is pretty much an equal distance to that. So what it's doing is kind of like basically like drawing a ball around it and then drawing 3 axes through the ball, the 3 radii. And then it's flattening that. So that's why when you see here, for example, like this basically PC1 versus PC2. It's like you're taking this ball, you're rotating it to explain the most amount of variance, and you're just pressing it onto 2D. So, you can see that this red dot here is now like next to these yellow-green dots. It's not in real life, but in principal component 1 versus 2, it is. And it's just because the way the data varies spatially is it's almost evenly varied spatially. But in terms of the way it varies relative to its nearest neighbors, it's very different.
But I think what I need to, how often do you guys have these work? This is my first time coming to the workshop. I think I need to sit my butt down and probably go through this, but how often are these hosted?
I think every week. This is my first one, to be honest.
So you can probably find the tab there, but they're also going to play it around. But if you want to, I can send you a link.
I would appreciate.
Yeah, what's your name?
My name is Laura. Because right now what I'm doing is I'm mostly meeting with Jocelyn once a week, and she's helping me out.
Oh, yeah, Jocelyn knows about this as well.
Yeah,
Just
that's
all
all.
of you.
Yeah. So that's awesome. But I'm kind of like
Thanks, man. Oh, I see there's a question.
What was your break?
It's good. Sorry, just give me like 1 second here. I do see there's a question in the Zoom. Yes, sorry. Rijang, curious about the following question. After we've fitted a t-SNE or UMAP and we want to apply these low dimensional maps on new data, can we do that? And if yes, what's the inference time? I'm not sure what you mean by the inference time, but yes, we can absolutely do that. So what you can do is, and I think I have the code in the notebook, but basically you just save the object. So when you do like umap.fit, you can save that as an object, and provided the original data is the same dimensionality as the new data, you can just paste it onto there. So I have a paper, my 2019 paper that's linked in one of the notebooks. That's actually what I did with human genetic data, where I created a basis using UMAP, and then I projected new populations on top of it so I could figure out how these were related to each other. You can do the same thing in principle with cell types. So it is in there. So you use the, and just to put it in the chat, you would use .fit, and then later .transform, I believe. I'd have to double check that, but I know .fit is what you would do. And then from Catherine, could you explain more about the elbow topic? How is it in some sense principled, in some sense arbitrary? Right, so the scree plot, it's principled in the sense of like, well, we have a measure. That's kind of what it is, where you want to maximize, you might want to preserve the top principal components such that the new principal components don't add significantly more information than before. So it's principled in the sense of like, all right, we're going to take the top 10 principal components because they explain, say, 85% of the data. And after that, everything else explains like 0.15% of the data or less. Maybe it's not useful to you. So it's principled in that sense that you do have a like numerical threshold you're using. And you can also do a hypothesis test on the significance of eigenvalues using the, I want to say, the Tracy-Whittem distribution. Now, on the other hand, it is a little arbitrary because I've seen this in population genetic work. So you remember that scree plot where it had a huge drop off after 4 principal components. We see that a lot in population genetics. But in my research, I found that there's an enormous amount of signal in like the top 100 principal components. So there's still signal in the data. That signal might not explain as much of the variance. So it's one of those things where, in a pinch, if you're doing some sort of pre-processing for a pipeline, maybe 90% of the data is as much as you need. If you're trying to say like, oh, there's nothing significant beyond 90%, I would not necessarily go that far. Or, the top 100 principal components might only explain, say, 10% of the data. In that case, the scree plot would not be informative because there's still quite a lot of data in the remaining principal components that you would drop from the scree plot. Does that help? All right, cool. Thank you, everyone. Yeah, any more questions? Then I'll be free. I'll be happy to answer over email or something. Bye-bye.